Hello and good evening to everyone. And we're going to have an absolutely fascinating discussion because we're focused on mRNA vaccines, and critically, we're focused on mRNA vaccines in the context of someone who was the discoverer or the creator of it all the way back in 1988. That's Rob Malone. And we're going to explore some of his ideas, some of the history, some of his experiences. And I hope that you will find this as absolutely fascinating as I have. So here we have with us, Rob, how are you?
I'm good, and thank you, Dr. McMillan, for having me on your podcast, and for the opportunity to discuss ideas and data and science and everything else.
No, listen, let's get straight to the crunch point that I want to understand. You were a PhD student when you first came up with the concept of mRNA vaccination. Were you the first person ever to think about this?
I can't say whether anybody else preceded me in thinking about it. I'm not aware of documentation of others having done it. In my experience, when the time is right for a technology or a discovery, it tends to emerge kind of organically all across the world. In this case, strangely, it doesn't seem to have done so in that time frame of the late 80s. And so the record suggests that I was the first to develop this idea, but I can't say that nobody else had it. That'd be impossible.
And so when you think, because you were studying, you were doing your PhD at the Salk Institute at that time, and when you were working on your dissertation with mRNA, and just to clarify, in case people don't fully understand, even though we're talking about it in the context of COVID, a lot of people may not understand the simplistic perspective on mRNA. What really were you doing in a very simple way for somebody to understand?
So you're kind of asking the question, how did this all come about?
Yes.
And I can assure you that I did not start off thinking I'm going to have a PhD on RNA vaccines. Absolutely not. The genesis of this is that I was fascinated. I was fascinated with retroviruses. I had kind of cut my teeth on mouse mammary tumor virus, a breast cancer virus of mice, and in a laboratory that played a key role in the initial discoveries having to do with the role of retroviruses in AIDS back at UC Davis at the Primate Research Center. And so that was my background, and I'd come out of that as entering into an MD PhD program, absolutely fascinated with the idea of retroviral-based gene therapy. And I thought this was something that I could do with my fascination and knowledge of retroviruses. And that could be, and it looked likely that this was really going to happen technically. And it's something that I could continue to pursue through my entire career. And by this point in time, there would be gene therapists everywhere in every hospital. That was my vision at the time. And so I went to this university, UC San Diego, that had two of the absolute pioneers in retroviral gene therapy, Inder Verma and Ted Friedman. Ted had been the one that really came up with the whole gene therapy idea. Now where the rubber hits the road for the young graduate student is, what are you actually going to do? What is the question you want to work on? And for me, one of the core questions was, how does the RNA of the retrovirus assemble into live virus particles and get produced as viruses? This was essential to the fundamentals of making retroviral vectors work. And there was enough known about the RNA of the retrovirus and the specific packaging sequences that it was possible, theoretically, to examine that RNA, mutate that RNA, and ask questions about how those specific sequences we're interacting with cellular proteins and viral proteins to assemble the retroviral particles that would be produced.
Hey,
But
just hold before you go any further. Let me just clarify, because I'm talking really basic stuff for people. When you say RNA, usually the DNA in our body is what makes all the proteins. Well, it goes from DNA makes RNA, and RNA then makes the proteins.
Central dogma.
Yes, and so when you talk about RNA, you are talking about going in the middle between the DNA and the proteins to be able to change the way how a cell works. Is that about right?
So that kind of jumps ahead in time to the idea of mRNA as a drug and mRNA as a vaccine. Mm-hmm. At this point in time, I just wanted to set the stage. The truth of the matter is retroviruses, and it's captured in the name. Retroviruses are very odd, and David Baltimore got the Nobel for this, in that they have a protein that will take from the RNA and make DNA, which then gets put into the chromosome of the cell, which then makes retroviral RNA, which then gets packaged and put out as new viral particles. That's the life cycle of a retrovirus, is it's got this backwards step. But yes, you're exactly right. The central dogma of biology is that DNA makes RNA, RNA makes protein. Mm-hmm. So in this case, I was trying to ask this question about how retroviruses package RNA so that they can produce viable retroviruses. And in order to build that as an experimental system, I had to have a way to produce synthetic RNA with mutations in it, put it into a cell, and ask whether it comes back out as virus particles. Mm-hmm. And that's what got me going on this. It wasn't because I was going to change the world or make an RNA vaccine or any of that stuff. That's where it started. I was in an amazing hothouse gene therapy environment. Just to give an example, the senior postdoc, when he left the laboratory, created the leading adenoviral vector company that now has been sold to Johnson & Johnson, and that technology is the basis for the J&J vaccine now. So I was in this amazing pressure cooker doing these gene therapy experiments, and surrounded by people grappling with the early days of gene therapy and what the problems were.
Was there any specific moment when you thought, oh my goodness, this is a possibility to use this technology to make a vaccine, all the way in the 1980s?
So there was two kind of events. The first one was the aha moment about using gene therapy technology to produce vaccines. And that happened because I was working very closely with a kind of a postdoc mentor whose name is Dan St. Louis. He still lives in San Diego. And Dan was working on a system where he would use retrovirus vectors to put genes expressing a secreted protein into cells of a mouse, and then collecting them in a little package, a nodule, we could say, placing them as a transplant into the mouse, and then the mouse would continue making the protein. Okay, so this was putting things into engineered cells in cell culture and then implanting them back in the mouse. That was the model, using a retrovirus. And Dan ran into a problem. It was a huge paradox in the laboratory. The cells would produce the protein in the mouse for about 3 weeks, and then they would stop producing it. And Inder's lab, where I was, Inder Verma, one of the top leaders at the time in regulation of gene expression, and the others who were supporting him in that mission, thought that this must be what was happening was some sort of a control event, that the retroviruses were getting turned off in the cells. And the aha moment for me, as somebody trained in medicine for a couple of years and immunology and vaccines and all that, was good heavens, this is happening at 3 weeks, which is exactly the timeline for a good, robust cellular and humoral immune response. Probably what's going on is not some epigenetic, fancy chromosomal silencing thing, but rather simply that the mouse is making an immune response against the foreign protein.
Mm-hmm.
And lo and behold, that was what happened. Now, it seems like a very small thing in retrospect, but it was heresy at the time. Gene therapy had not yet come to confront the fact that it has this fundamental logic flaw, which is if you're putting the good gene into a patient, the patient's immune system doesn't know that it's the good gene. It only knows that it's a foreign gene, and it will mount an immune response against it, and it will also mount an immune response against the vector if it's an adenovirus or a retrovirus or whatever. Okay, so this aha moment seems trivial in retrospect. At the time, it was a kind of heresy and not well received, as you might imagine. But that was one of the key ones. Was gene therapy can be used. It may not ever. Remember, I come into this a true believer in gene therapy. This is going to be my career. Okay, I'm all in. Okay, and suddenly the epiphany. Oh, this isn't going to work so good. What are we going to do about it? Right? Because if you've got an immune response, there's no going back. Then you're into immune suppression land and really complex science, and that doesn't work very well. But the aha was, one of the way out of the woods is that we can use gene transfer technology to make vaccines. And so that was a key aha. And I thank Dan St. Louis for helping me and mentoring me at that time when I had that brainstorm. Wow. The other one that was kind of pivotal was way downstream and happened because of this serendipity that I was working with a non-viral method for transferring RNA into cells. And then because I was working as a teaching assistant in an embryology course at UC San Diego, and I had extra Xenopus embryos. So these are basically early stage tadpoles. And I took the material that I had been using for cell culture to test. I was testing a whole large array of cell culture lines for the ability to put RNA into them. And I said, well, let's just see what happens with these frog embryos. And those RNA experiments worked to my great surprise, astonishment, really. They worked incredibly well. And then we, the next stage in the embryology course was some experiments involving chick embryos. And so I did this kind of same thing. Well, if it works with frogs, what about chicks? And lo and behold, I got signal with chicks. And so at that point, then things got serious and interesting. And disclosures were filed, and people started wrestling over who got ownership and all those kinds of things, and it all went sideways.
So this is where you
with these embryos and how well this worked, and it was totally unexpected. But once there, this was a simple 2 + 2. It doesn't work that well. I'm not going to cure cystic fibrosis or muscular dystrophy, but it might work well enough to make an immune response. And that was kind of the synthesis.
So listen, now this raises an important phase, because at this time you were also having difficulty as a PhD in that environment, and you did actually leave before you had finished. Was that a difficult time for you personally?
So I ended up with a diagnosis of post-traumatic stress disorder from a university physician. I went through some experiences that I wouldn't wish on my worst enemy. With the perspective of time, I would not be who I am now if I had not gone through those. And I was absolutely an aggressive, ambitious, overconfident young graduate student. You mentioned before, if I could speak to my younger self or to other graduate students, I would tell my younger self a story about the importance of humility and being a little bit more low key about things. But that's who I was then. It's not, the experiences profoundly changed me, and I'm sure for the better. But they were extremely painful going through.
And so let me ask you a hard question on that point, because you left the Salk Institute, and in a sense, you left a lot of that work behind you. Do you think that you'd be in a completely different place if you had stayed and continued with that work there?
Okay, so probably not. The kind of cascade of serendipity continued when I left Salk and joined a little startup working for a scientist that I'd been collaborating with at the company Syntex in Palo Alto, that had been hired to this La Jolla startup called Vical. And what happened was that I brought the protocols and reagents and know-how into Vical, and there was capital and willingness to support further animal research and to take it to the next step at Vical, and that was what led to the science paper that was, considered to be analogous to cold fusion. That was the naked DNA and RNA paper. That wouldn't have happened had I remained at the Salk. Absolutely. Would it have been better for me in terms of my long-term career trajectory to stay at the Salk? I don't know. It was impossible for me to remain at the Salk psychologically. It was not an option. I had in the context of all this recent churn about those old days, a colleague, another postdoc from the lab, wrote to me personally and empathized, said that they hadn't realized all that I'd been going through, but they shared what they'd gone through and what they had seen other people go through. And you may or may not be aware, I don't think I'm sliding. Dr. Verma was asked to leave the Salk Institute, where he was basically running the place, after an expose in Science magazine about sexual harassment, which this individual that wrote to me privately reminded me of some of the things that he'd observed. So, and he spoke about Dr. Verma's practice of being very casual with the intellectual contributions of others in the forms of manuscripts, grants, etc. And the individual had the same exact experience that I had, where Inder would, at one point Inder gave me a stack of grants that he had to review, and he said, read these and get ideas. I was shocked. And I went to the then president of the Salk and said, this isn't right, is it? And the response I got back was, well, maybe Inder isn't the best role model for you. So there was kind of a tolerance there for a number of things. And I was an ambitious graduate student in a postdoc lab of, 16 postdocs or something that were extremely competitive. And the practice was to assign multiple postdocs to the same task and see which ones succeeded. So you can appreciate it's just a pressure cooker. Yeah. And I was warned not to go there. And yet I chose to do so because I was so passionate about gene therapy and what I wanted to do in my career. I can't, I brought this upon myself in some ways. Maybe that's speaking, a victim, but regardless, it was not a psychological option for me, having been this starry-eyed, naive, ambitious young scientist, to remain in that environment that I had walked into, thinking it was the temple of knowledge. With, Francis Crick was there at the time. There was a half a dozen Nobel laureates. It was kind of the center of the intellectual world of molecular biology, that and the Whitehead. And so in retrospect, can I say I should have done it differently? There's, one makes many mistakes as a young person. But, were they useful or not? I think that has to do with what you do inside with those experiences. And hopefully I've processed them, and I take some comfort that I have had a long series of contributions since. But one of the things that comes out of that is I am really religious about ensuring that people get credit for their contributions. And I think it makes me a much better leader.
So that raises a question before we move on in the discussion, which is that when we see all the attention now on the success of mRNA vaccines and so on, how does it make you feel, considering that you first, I guess, documented the idea, it was your baby, you were taking it forward, and then you almost had to walk away from it, and yet you are not giving any credit for it? How does that make you feel?
Okay, let me frame it up a little bit. I had no choice but to walk away from it, because Vical, in my employment terms and agreement, had employment terms there that made it so that everything I did at Vical was owned by Vical. And furthermore, I was not allowed to continue to develop that line of research. It belonged to Vical, and they sold it to Merck. So for many years, all of this data and information that I've recently, and my wife has largely disclosed, was hidden away in a very large box in our closet. And it wasn't an option. Now, Vical recently went bankrupt. And, as a young academic, I received cease and desist letters from Vical. So it really wasn't an option to carry on in the same way. I had to do new things. So just to get that, the feeling, so Francis Delier of Heidi News was the one that first contacted me from the press about this whole situation. And from the heart, what I said to him was, it feels like rape. And I know that's a strong word, but what I was trying to express is it feels like something that I have created, and with my own passion and effort. I mean, I worked tirelessly. Is being taken from me completely, without anything, being taken by others, and that's pretty rough. In academe, there's rules about citing, citations. You know that, you're an academic and you publish also. And it's probably pretty important to you, as it is to me, in this small, strange world of academic research and publications, that people acknowledge your intellectual contributions. And I'm sure you, like me, are rigorous about acknowledging the best metaphor is we all stand on the shoulders of giants. Mm-hmm. And I think it's important to acknowledge those that have contributed to this song that we all sing together, that have come before us and are our peers. And to have that kind of taken away, and others seeking to advance their own agendas without even acknowledgement is a little rough. So how does it feel? It hurts a lot. a quote, I think it's from the Outliner, you know, it ain't right, it ain't proper. And I think we all know that, right? Intuitively, but science, in academic science, is a strangely competitive world that's very much a pyramid.
Mm-hmm.
And so it's a personal commitment on my part to make sure that everybody that is in my world gets credited for their contributions. And I guess I kind of expect the same from others regarding my contributions. It's the only thing. It's part of the capital that we're all trading as as academics and intellects, contributing to this common song that we sing in science, and that we will all respect the contributions of each other. Have I answered your question?
Absolutely, absolutely. And I feel honored that I'm in a position to be able to at least give you the opportunity to get some of that credit, and
That's kind.
I feel honored about that. So I'm going to take us on to a different part, because we are in the middle of COVID. You are therefore one of the experts on mRNA vaccination. And it has done a tremendous step with regards to reducing.
I'm shocked. I mean, I am amazed. I spent a couple of decades of my career trying to advance new gene delivery technologies. I was a founder of Inovio, the electroporation company, and mentored the young Norwegian scientist that was behind that. I spent years and years developing and testing cationic lipids. I know how hard this business is to get this level of transgene expression, because that's really what it is. This is really gene, back to the original concept. This is gene therapy applied to vaccines with RNA. Wow. I thought, given what had happened and all the pushback and all the intervening time, I thought that this would eventually come to pass, but I would be long in the ground when it happened. So to see it happen so suddenly in the context of this amazing public health crisis we're in is mind-boggling. And furthermore, the levels of activity of these lipoplexes, that's the technical term for these small nano aggregates of RNA and lipids that are formed, which are what allows the RNA to go into cells and be produced into protein. The level of efficiency associated with those is mind-blowing to me. And in particular, I think the unsung hero in this song here is Dr. Peter Cullis and his team, who developed this advanced new formulations of lipids that are directly being used by both the BioNTech Pfizer product and the CureVac product. The Moderna product is a slight variation on the same. The activity of those is mind-blowing. So I never expected to see this in my lifetime. And I'm fascinated, but with that high level of transfection activity of, polynucleotide delivery ability, there comes consequences. As you know, in drug development, nothing is free, and no drug is perfectly safe. And so with this amazing advance in efficiency come some potential issues, which I think we need to be sensitive to.
Now, this is a sensitive topic because, as I said, we're in the middle of a pandemic. There is a lot of pressure politically to try and get out of it with regards to vaccines, and a major part of it, especially with some of the other side effects that we're seeing from some of the other vaccine candidates, it looks as though the mRNA technology is leading the way. So you've made a good point, is that there is the opportunity to make a difference, but as always with any technology, there are risks or potential problems. This is an important topic. What have you come across recently that would make you think about consideration of how we approach vaccination at this point?
So thanks for that. And let me frame that up a little bit. I was alerted from a colleague that worked for the government, let's say US government, that was in Wuhan during the last month of 2019. I was alerted in the first week of 2020 about the threat that the virus represented. And I made a threat assessment. I'm an experienced outbreak specialist that does a lot of biodefense work these days, mostly with the US government. And so I made a threat assessment and made a personal determination that the most efficient way to help provide protection was to focus on drug repurposing, not on vaccine development. Now, the government of the United States made a different assessment and different decision, and I don't want to second guess that. It's water under the bridge, and I can tell you, having been through too many of these outbreaks, when it hits and the fog of war hits, it becomes very difficult to process information and make decisions. This decision was made by the government to pursue vaccine technology because there was apparently, by these emails that have come out recently from the Washington Post under FOIA, there was a sense that this was the best way to move forward. It was to invest all capital and effort into vaccine development, and the NIH, as well as BioNTech back in EU, made a strategic assessment to invest in RNA-based approaches, which was quite bold. And that decision has been validated. And so I've watched with great interest as this has proceeded. Recently, because of my background, and I'm a clinical development specialist, I'm experienced in toxicology and regulatory affairs and other things. This is what I do for a living these days. I was approached by some individuals to provide an assessment, an independent assessment of some data that had been revealed through a Freedom of Information Act submitted by a group of Canadian scientists and physicians, which had obtained a copy of the application which Pfizer and BioNTech had submitted for their mRNA vaccine to the regulatory authorities in Japan. And this is forgive me, there's some technical words here. This was their initial new drug application, or their common technical document. These are regulatory terms. What it means is it's the information package that a drug developer or sponsor, in this case vaccine developer, would submit to a government regulatory authority that basically says, here's the sum total of all of our data at this point in time supporting the use of this product in humans. And so normally these packages of information are closely held confidential and are not discoverable by Freedom of Information Act. They're usually protected from that, and that's certainly the case in the United States. But these Canadian scientists and physicians who were very concerned about some of the safety signals that they were seeing in vaccine recipients in Canada decided to, of their own initiative, to seek more information. And so they obtained this, and within 24 hours, I was asked by the editor-in-chief of an organization called Trial Site News in the United States to look at this document and another document that was publicly available from the European Medicines Agency that was their official summary of their assessment of the analogous initial new drug application and common technical document that had been submitted to them.
Can I stop you here a minute, Robert, before you go any further? Because we're speaking live, and this information is being shared publicly, and you are walking in an area here that is not well taken, where you are saying stuff that can be challenging to the political narrative with regards to vaccines. Are you sure you want to speak about this? And
Yeah, so thank you for giving me the opportunity to pause and reconsider. I'm really committed to openness and transparency, and part of that comes from the background of the crucible that I've had to go through, but it also comes from the extensive training I've had in bioethics, and I'm personally, as a physician, scientist, clinical development specialist, I have a really bedrock commitment to fundamental principles of bioethics. And among those is transparency and the importance of full disclosure to particularly people receiving experimental products. So I'm comfortable continuing to speak. I'm an experienced professional, regulatory professional, and I hope that the audience doesn't in any way infer that I am invested in a contrary position regarding vaccines. I'm not an anti-vaxxer. I've spent my whole life developing vaccines and trying to advance vaccines, but that doesn't override my commitment to transparency and disclosure and good science. I'm of the belief that the adult public is mature enough to make their own informed decisions based on the information available, and that it's the obligation of governments and public health organizations particularly in the context of an experimental product like these vaccines all are right now. It's the obligation of the public health community, the official public health community, to convince the public that they're safe and effective. So I'm totally okay with proceeding, and I'm a grown-up too. And if I say something that's controversial and I start getting hate mail, it's part of the price I have to pay. But it doesn't. I've been through situations before where I have gone public about major ethical issues that I've encountered in this kind of technology development space and drug development space and clinical research space, and I've had consequences in my career, but that doesn't absolve me from the obligation that I think I have to be open and transparent about information which is relevant and, in my opinion, should be made publicly available.
Well, that is tremendous, and we do appreciate that, because we do need scientists like yourself who are studying and able to challenge and make sure that everything that we do is as safe as possible. So yes, please go ahead. We are very appreciative of that.
So thank you for that support and those words. Let me just kind of loop back just a little bit on that theme. In my experience and my strong opinion, science requires that we challenge each other. And if we don't do that, we end up with groupthink and we end up making mistakes. So continuing on the theme, these documents were provided to me and I was asked to render an opinion about what I saw as a professional with the level of, the type of experience and background that I have. These documents that had been uncovered by my respected scientific and clinical colleagues in Canada. And so I made an independent assessment of those. And then I was concerned that I may have overinterpreted or misinterpreted. And so I asked an even more senior regulatory specialist that I respect highly to look over the same documents and draw their own independent opinion to make sure that I wasn't overreading things or overreacting to them. So that happened. And with those documents, we both observed that there were deficiencies that were highly unusual and not what we would anticipate seeing in a regulatory submission from an organization of the experience and capabilities of Pfizer. Mm-hmm. That was, I found that a little bit alarming. So did my colleague. He was less sanguine about placing his name to his assessment. And so he remains anonymous. But I was comfortable that the assessment that I made was true and accurate based on the documents that I had. And I was very clear on that. So the findings included that there were data in the IND package from Japan, data tables that examine the biodistribution of the vaccine product in a animal model, in a rodent model, with limited numbers of samples using two core technologies. One was the one that I used way back in the day when I was pioneering RNA delivery, which is expression of proteins from an RNA encoding the protein that makes the firefly tail glow. Okay, so it has the perhaps unfortunate name here in the States, where we have a strong religious community, of being Luciferase. Luciferase is just the name that the discoverers that were trying to understand how come fireflies glow at night gave to the protein when they cloned it. And it turns out to be a super duper, we call it reporter protein, way to detect whether or not a polynucleotide, an RNA or a DNA, has gotten into a cell and been turned into protein, because it will produce photons, and those are easy to detect. So a lot of technical there. So the studies were done in non-good laboratory practice means, and they involved use of the luciferase RNA rather than the actual RNA of the drug product encoding spike, and they involved tritiated RNA. So this is radioactively tagged RNA that is able to be detected very sensitively because it's radioactive in various tissues of the animal in which it's been injected. And so the data that I was able to review had to do with the distribution of injected RNA lipoplex complexes. We talked about that earlier. This is the actual material, only it wasn't the actual material, which is what usually one has to do these studies with. It was a surrogate, the RNA for luciferase, instead of the RNA encoding the vaccine, which is encoding spike. So what the data showed was that in these rodent models, and it was small numbers of rodents, that the injected material, a lot of it stays near the injection site, but a lot of it doesn't. And it goes throughout the bodies of these mice or rats, and it appears that it may have remained biologically active in some of those distal sites. And it certainly, the data suggested, and the tables are a little difficult to follow for me because they analyze both the lipid distribution and the tritiated signal distribution, which is a marker of the RNA. They showed a high concentration and relative concentration into, for instance, ovarian tissue, spleen, and liver, but in particular, the ovarian tissue was surprising. And particularly, it was surprising because there was not that concentration in the testis that were sampled from the male rodents. Okay, so we had concentration of the lipids in particular in ovaries, and we had some relative concentration of the tritiated signal suggesting the RNA in the ovaries compared to some other tissues. But strikingly, we had distribution of all of this kind of systemically throughout these animal models.
Is that what we would expect to a certain extent with regards to injecting a vaccine? Isn't there some degree of spread, or do we expect it to remain local to where it was injected?
Good question, and entirely appropriate. I had no way to assess, because we didn't have the controls of alternatives. But yes, one would expect to some degree a spread of the injected material. The core thesis that had been advanced in the scientific literature before I can only speak of my personal experience, my encountering these data in this IND package, IND being the abbreviation for initial new drug application. My expectation from the literature was that the injected material and the encoded spike protein would remain membrane bound and regional. I was aware that these complexes had been engineered as a formulation historically, specifically so that they would be transmitted along lymphatic system. And I'm trained in pathology. I used to teach university pathology, and I know darn well that the lymphatic system drains into the blood and circulates and passes through the liver and all that good stuff. This is, fundamental biology and human physiology in mammals. So the prior publications and information had suggested that these complexes would remain at the site of injection or the draining nodes. The formulations were specifically engineered to preferentially go through lymphatics to the draining nodes, lymph nodes, which is where the immune system processes antigens and teaches B cells and T cells. These are the things that attack other cells or produce antibodies. Teaches them, educates them so that they make the proper immune response against the antigen, just for the viewership. So that was the logic. Spike was engineered with a tether. It transmembrane region. It was supposed to stay in the cells that it was that were in the region, and it was not previously disclosed, let's say, in the literature, that it would move systemically in any kind of a systematic way, significant way. So
can I
for
ask
me,
a
it was a
question
surprise, and the relative concentration in certain organs was a bit of a surprise. You'd expect it to concentrate in spleen and in liver.
Yes.
The ovarian signal was a little surprising, more than a little surprising, a bit concerning, particularly since now we get into this intersection between these many reports that patients that are out there, that patients have made, and we have to be really careful because there's a selection bias. When somebody makes a spectacular claim, I lost my baby, or I had, my parents died, or, right after vaccination, or whatever. These things quickly catch fire in social media these days. And the truth is that all of these events would be expected to occur at some normal frequency. And for instance, there are those who say, we have X number of vaccine-associated deaths. Well, And then there's others that I've heard, public health officials, saying, no, there's no vaccine-associated deaths. Okay, well, I know that's false. Because by random chance, a certain number of people will die within the 2 weeks after they receive a vaccine. The question is there any causative relationship between those? And is there more people dying at, for example, just to take the extreme, are there more people dying than would normally happen by random chance in X period of time after they received a vaccine? Those are the key issues. And so there's various claims that are made, and people are, there are those that are very upset and understandably so about risks, including this group of Canadians. And I hear about it all the time, as you might imagine, with my background. People on the street, the house cleaner, should I take a vaccine? I have these problems, this background. And I'm asked to opine. So I get hit with this. In this case, some of these reports about dysmenorrhea or other ovarian-related events that may or may not have been underreported by public health authorities, or may have been readily discarded as not vaccine-related. Because this is all subjective. That's the one thing about this. We can talk as if this is hard science and we're all dealing with numbers and statistics and machine learning and all that wonderful stuff. But the truth is, it comes down to somebody making a subjective decision. This is related to vaccines or it isn't related to vaccines, because it's, and that triggering an investigation. So that, if the person making that decision, that call, has any intrinsic bias, this can cause problems, and we're very aware of that type of problem in clinical research.
I want to pause you here a minute. I want to take you back to something that you'd said, which was in relation to the spike protein spreading systemically, that's mean throughout the body. I remember seeing a very interesting study in Israel where they were looking at patients with hematological malignancies. And they realized that they can't do PET scans within a certain time frame, because if they did, they were starting to get false positives because nodes were lighting up all over the body and they couldn't differentiate post-vaccination. Does this fit with spike protein going and spreading into nodes and spleen throughout the body?
Okay, so we have to allow me to dissect that a little bit, and it's a fascinating observation and insight that you're sharing, and thank you for sharing it. It's adding value, and this is a great example of how good science happens. Is a lot of the folks, it takes a lot of minds, the expression, it takes a village. It takes a lot of people thinking really hard and looking for associations to pick signal from noise. And you've just given a great example of that. So let's pull apart a little bit what you said. We're speaking about spike protein. So the data that kind of lit me up a little bit with looking at the Pfizer IND package was relating to the distribution of the transfection complexes. Transfection being a fancy scientific word for the process of the RNA getting into cells. I was talking about the data relating to the distribution of the lipoplexes, which cause the RNA to get into cells and turn into spike protein. You're talking about spike protein. And so there's another key paper that I'll remind you of, I'm sure you're aware of, that recently it's in press, it's past peer review, and it's in press from Harvard and Brigham. So generally, this is like publication in NEJM. If it comes out of Harvard and Brigham, we all say, oh yes, that must be true. But in any case, it was a bunch of nurses, a group of nurses, about 14 or 15, that received vaccine and agreed to Moderna then in that case, and agreed to have blood draws after vaccination over a period of time. And some very sophisticated, sensitive technology was used to detect the presence of free spike protein or subunit in their blood. So in that data set, they're looking at the actual protein, which is what you're referring to in your example. Mm-hmm. And of course, the production of the protein is a little time shifted. It's a little delayed compared to the distribution of the RNA. And the engineering that had been done at the Vaccine Research Center for the Moderna product and at BioNTech for the Pfizer product had engineered the spike so that it would stay put where it was expressed. And yet these nurses had significant levels of non-trivial levels of circulating free spike protein in their blood for a prolonged period of time. Now, for me as a gene delivery guy, this is kind of, wow, I'm shocked. It's amazing. I can't believe it. Well, not really. I mean, I believe the data, but it's profound.
And I mean, I guess profound in a good way or a bad way?
In terms of the underlying technology, so both. Like we said at the beginning, there's a yin and a yang to these things. So as a technologist who had worked for decades trying to develop more efficient systems for non-viral gene transfer, this was shockingly good. As a trained pathologist, clinical research specialist, physician scientist, this was Houston, we've got a problem, right? Because this isn't supposed to be happening. Mm-hmm. Because spike protein is biologically active. It's not an inert antigen. It's not just an antigen. The virus has evolved to do a whole host of amazing things. And these little RNA viruses are just evolutionary pressure cookers. And there are all kinds of overlapping activities that exist in all of the expressed proteins for these little RNA viruses, like the SARS-CoV-2. Mm-hmm. So spike is not just an antigen. Spike has its own intrinsic activity. And as you note, among those things is that it binds to a particular receptor present widely in humans, particularly in vascular endothelial cells. That's a fancy word for the cells that line the inside of your blood vessels. And they express and display this ACE2 receptor. And binding of ACE2 is crucial for regulation of a whole bunch of things, including blood pressure. Mm-hmm. So if you have something in the blood that can bind to ACE2, it can cause all kinds of issues, many of which we're only beginning to understand. So what that also means is that the free protein detected in the blood of these nurses is a tiny subset of the total amount of free protein, because most of it is probably sticking to ACE2 on vascular endothelial cells, among other things.
Okay? No, this is a very important point, because my research is focused on the autoimmune response in COVID-19, and the fact that what I have been predicting is happening is that the serum, or the free-floating ACE2, binds to the viral spike protein, and that combination then triggers an autoimmune response. And from a clinical perspective, what I've noticed is that many patients have symptoms about 2 weeks after the vaccination, which would fit with an IgG production time, as opposed to necessarily being directly related to the spike protein. So are we triggering an autoimmune response as well as an immune response to the viral spike protein? Is that possible?
Thank you for that last caveat. Is it possible? Absolutely. We're in so this is science, right? This is science, the intersection of science and medicine. And I'm a big fan. As a very young undergraduate, I was taken under the wing of an experienced pathologist at Davis. And it was a little bit of a brutal experience. I got a lot of things drummed into my head. One of them was this ancient paper from the 1880s, published in Science, called The Method of Multiple Working Hypotheses. And it's how I've approached science ever since. So the core idea is, don't get invested in just one hypothesis. Recognize that when you encounter something scientifically, some phenomena, which is what we're talking about, then it's really good to come up with as many possible explanations as you can think of, you and all your buddies, and then design experiments to reject those so that you get down to a core of hypotheses that you haven't been able to reject. And then there's a good chance one of those is the true thing. Now that's different. Some people have a tendency to say, aha, I know what the problem is. It's this. And they'll go and spend a lot of time focused on that. But I think there's a lot of merit to this idea that you are pursuing of autoimmune. And absolutely, I'm a fairly knowledgeable immunologist. And I've spent time in computational immunology and cellular immunology, et cetera, more cellular than humoral for me. These antigen complexes, which is what you're talking about forming, whether they're membrane associated or free, represent, again, it gets back to the fundamentals. The immune system doesn't know that it's you or not you. They just know whether or not it's different from what it's seen before. And anytime you do something like this, where you have a foreign protein complexed with a self protein, then as far as your immune system is concerned, that's something different. And it needs to respond to it. And we have, our knowledge of the immune system is still evolving. And these days we have T regulatory cells and all kinds of complex regulatory networks. But is it conceivable that that much of the delayed pathology observed in COVID disease, recognize that COVID disease is not the same as SARS-CoV-2 infection? A lot of people get infected and they don't develop clinical disease. Only a subset do. And often that more severe disease, the part that puts you in the hospital and kills you if you are unlucky, or puts you on a respirator with a lot of oxygen, that often is delayed, as you say, by it's hard to measure because we're not subjecting humans to challenge studies right now, so we don't really know what the timeline is here. But it looks like, as you say, it's 2 to 3 weeks. Now, I would say one thing to your hypothesis about the rise of the IgG. Just forgive me, but I have been tracking this really carefully, and I completely agree that the autoimmune hypothesis is one of the leading hypotheses for. Here, just a minute. Somebody's trying to call me, and I'm going to hang up on them. 1 moment. One of the leading hypotheses for both the pathogenesis of COVID, the disease that's often delayed, severe disease, inflammatory disease, and what the community calls long COVID, the thing that I experienced, this prolonged syndrome that has a lot of symptoms that are a little bit inexplicable. That there are many different systemic diseases that are consistent with that. POTS is one of them. But much of the myalgia, arthralgia, and potential coagulopathy that's observed is consistent with autoimmune functions. Another one that's worrisome is the thrombocytopenia that pops up.
Yes.
My colleagues within the FDA, that I'm not going to share their names, tell me is a known complication of any oligonucleotide-based therapy in a small subset of patients, thrombocytopenia. So so the formation of these complexes is absolutely could yield a autoimmune response in a subset of patients. And let me amplify that a little bit because it gets back to the safety. As a vaccinologist.
But I want, yes, this is a bit that I want to try and
Usually
clarify.
we don't even get close to administration of market authorization. That's the technical term for licensure for a product, for a vaccine product, until 1 to 2 years after at least 3,000 subjects, that's people, have been exposed to the product. Why is that? Because of autoimmune disease, largely, and the autoimmune disease risk, because these are often fairly rare events. Now we can, I can't venture an opinion right now. It's not my job to examine the data about whether or not these are rare events or relatively common events that we're seeing. But the regulatory agencies throughout the world generally agree that you want to be able to detect a severe adverse event, which autoimmune is an example, at a frequency of about 1 in 1,000. Okay, when you power your clinical studies. And the statistics just work out that if you want to make sure that you detect something that happens 1 in 1,000 patients that receive the product, you have to expose 3,000 people. And then on average, you would see 3 people that had that event. So it would come across as a very rare thing. But if it's Guillain-Barré syndrome, paralysis of the face, that was observed with the swine flu vaccine and is part of the safety signal associated with flu vaccines in general, it occurs at a higher rate than that. So usually, because autoimmune diseases manifest over a longer time course, regulatory dossiers, regulatory agencies require that we have 2 years of safety follow-up data after administration to at least 3,000 subjects.
So in this context, I'm
right
pushing
now,
you on this,
we're in a
this
situation
question.
where these things have not been deployed for 2 years.
Yeah.
We don't have those data. The safety data are emerging, but we also have, I like to say the wolf is at the door. People are dying. They're filling up our hospitals, fortunately not at the same rate that they used to. I'm watching the UK carefully to see the impact of some of these new variants on disease attack rates and incidence in the UK. They don't seem to have come stateside in quite the same way yet. I work closely with colleagues in India that are developing vaccines, more traditional ones, and that is a very unpleasant situation right now.
So here's a question.
So
Rob.
I hope that the UK doesn't experience something similar and that we don't see that in the States, but time will tell.
So Rob, I'm going to have to ask you here, based on what you have seen so far, do you think it's appropriate for us to continue with mass vaccination when we have these questions arising, especially in groups who are at low risk for severe disease?
Okay, I'm going to not give you an answer. Okay, and it has to do with me as a professional respecting my professional colleagues whose job it is to make that determination. Just to provide context, I did speak with insider colleagues that are very senior at the FDA that I've known for many years and trust, and they trust me, about these potential safety signals and concerns about widespread distribution of the gene expression and the high levels of spike protein free in the blood, and the Pfizer data package issues that I had observed. And they had recommended to me that I speak to the director of the Center for Biologics Evaluation and Research at the FDA. And he kindly granted me a meeting over Zoom about a day ago. And we discussed these various issues. He basically presented to me what he could, because he's constrained by his ability to disclose information associated with the Pfizer package that was fully submitted to the US FDA about 2 weeks ago.
Mm-hmm.
And I respect, he has an excellent reputation within the FDA as a person of intelligence and integrity. And so I trust that what he tells me reflects that that background of intelligence and integrity. And he assured me that the gaps that I had identified in the what are now really historic documents, they're from a different time frame that were obtained by the Canadian group, and may or may not reflect the current documents that have been submitted to the FDA.
I understand all of that, but we are at certainly in different parts of the world. There is a significant amount of pressure, especially now that there has been some, certainly more, they're aiming for the 12 to 15-year-olds. So children who are at low risk of severe disease. And what I'm saying is I do understand that there is due process, but this is in the context that if this is relevant for long-term impact on the young people, this is a pretty important point that has to be raised to the public very quickly. They should know about these issues.
You and I are aligned, and this gets back to the bedrock bioethics. Fundamental principles of bioethics are, for an experimental product, which is what these are currently, certainly in the United States, they're under emergency use authorization. They have not been approved yet in the United States. I can't speak it's a little unclear to me about the status in the UK. But I firmly believe that I'm of the school as a physician that your patients have the right to understand their disease and understand potential interventions and participate in decision making. It's their body. Your body doesn't belong to the state. And I'm of the opinion, as you suggest, that full and open disclosure of risks is something that not only can adults process and manage and should be able to, but I think we're obligated to have that full and open disclosure. So you speak now about adolescents, and that's a special case bioethically. If the product is not yet licensed and proven safe in an adolescent or child population, they, by definition, they are not able to provide full informed consent. They're not of the age of consent. This is fundamental. And in the specific case, there are now known disclosed risks in the adolescent population, particularly cardiomyopathy.
Mm-hmm.
Not so long ago, there was strong denial that there were any risks in this population, and that was kind of the group consensus and the party line, forgive me, put out by many public health professionals. But among other things, the FDA has a absolutely super duper biostatistician, one of the world leaders. He actually is employed by Oracle, and he examined a lot of those data. He discovered that there was a safety signal associated with cardiomyopathy in these mRNA vaccine recipients. That was then confirmed by the CDC. The CDC issued a press announcement and an announcement on their website. I received a document from the Maryland Board of Physicians, which is my licensing board, that specifically warned about cardiomyopathy in adolescents. And then we have an editorial in Science Magazine revealing that, everybody's kind of been relying on the Israeli database as the most comprehensive, and much reassurance was taken from the Israelis not having reported anything having to do with the adolescent population. And then they confirmed that, in fact, they saw the same safety signal. It doesn't inspire a whole lot of confidence, I'm afraid. And it's in this particular population that we've all agreed deserves particular caution and care. Now, if I can speak just a moment about cardiomyopathy, because that's, as you know, among other things, associated with autoimmune disease. That we don't know that that's the cause. We're not taking the small number of children, adolescents that have experienced cardiomyopathy and doing cardiac biopsies, which is the kind of thing that's necessary, because we generally don't do that. It has a lot of risks associated with it also. But I just want to share one thing. Because I work in this biodefense space, at the time when the US government, it's an anecdote, okay, so forgive me, has to do with smallpox vaccine. And it illustrates kind of the situation, I think, quite well. There was a time when the US government was concerned about the risk of smallpox being released stateside as a bioagent, a threat. And they started vaccinating first responders and other healthcare givers. And they saw what they believed was an intolerable signal of cardiomyopathy in the vaccinee population. And I was tasked, I was working as under contract with the DoD at the time, the Department of Defense of the US. I was tasked with looking into whether this type of a problem had existed historically during the prior major smallpox eradication campaigns that had occurred historically. And in fact, the signal was there. Soldiers and others, young people in particular, receiving smallpox vaccine had an incidence of cardiomyopathy that was compromising their function. And it was considered an acceptable risk because of the risk-benefit ratio. And that's crucial in understanding the perspective of the public health community about these signals. The thing about the risk-benefit ratio is it's subjective. It's in the eye of the beholder. So there's no, there's no computer algorithm or machine learning whiz-bang to say, you've crossed a line. Now you're over the risk-benefit ratio. It's very much a subjective call. And it's very much a function of what is the risk of the pathogen in the population. So if you were to do a risk-benefit analysis for, say, an RNA vaccine, and you were doing it from the perspective of the Indian regulatory authorities currently, you would be probably, responsibly very inclined to tolerate quite a bit of risk in the product because of the risk of death and long-term consequences associated with this variant that's spreading in infecting people in India. If you were doing the same analysis from the United States currently, as opposed to, say, 6 months ago, the risk-benefit ratio would change. Can you appreciate this?
Yes, I understand.
So with this adolescent population, it is not my job or pay grade to second guess national public health authorities. What I will say is that I am confident in my position in bioethics, and that position is, while these remain experimental products not yet licensed or fully evaluated in terms of their safety, there is an obligation to fully disclose risks, ensure that those which receive the product are comprehending those risks, and that they have consented in some way to accept those risks which have been fully disclosed and which they have comprehended. This is bedrock bioethics. And In the case of an adolescent, we have all agreed that adolescents and young people, down to people are talking about 2-year-olds, are not able to provide that informed consent. It can only be provided by their by a responsible adult that's been designated, whether it's their parents or whatever the designation has to do with, the people that have accepted responsibility for decision making on behalf of those adolescents or children. And I don't think, while these are still experimental vaccines, I caution that governments and health and public health professionals that are making these decisions about vaccination of adolescents in an environment where the safety database is evolving, and as we've seen, I think they are treading on very thin ice ethically, and I really counsel that it's past time for us as a world community and as a public health community to take a good hard look at what we're doing here. In crisis, it's easy to convince yourself of the need for extraordinary measures, and history is full of examples where we have decided to intern ethnic populations or whatever because of perceived risk, or allow certain types of medical experimentation on populations. The Tuskegee experience in the United States is notorious. African Americans in the United States are still wary of participating in clinical research as a consequence, for good reason. And I kind of feel like we're in that same kind of ethical environment where we have to be very careful and cognizant of the decisions we're making, because in retrospect, we may regret having done so, particularly because I can say for me, it's self-evident that the safety database is evolving. Mm-hmm. I look forward to disclosures from our colleagues in the Scandinavian countries and in the UK that have a well-developed national health service with more rigorous methodical reporting. Unfortunately, in the States, all of our safety databases are based on self-reporting. And the consequence is we have neither a reliable, I'm sorry, I got to say it, numerator nor denominator. We don't know what the event rate is for these adverse events, the true event rate among all people vaccinated. And we don't really know the number of vaccine recipients in any kind of a rigorous way. And these adverse events are self-reported. Yeah. So I don't want to second guess national health authorities and their policy. It's not my job. It's not my purview. But I do feel comfortable raising a concern about the underlying bioethics and reiterating that we, in the West at least, we all can agree on these fundamental principles. For an unlicensed product that remains experimental, there has to be full and open disclosure promptly of risks to the patients in the broadest sense. When you're running a clinical trial, that list of risks that are disclosed goes way down into the weeds, just like it does in a package insert. Yeah. And so we have to disclose. I think we're obligated to. We have to ensure that the public comprehends that disclosure. We have to put it in plain words that they can comprehend and not obfuscate it. And we have to have a process in which they are consenting. I believe firmly the state in the West, at least, does not have the authorization to compel vaccination or administration of an experimental product, full stop. And I'm not shy about saying that.
Absolutely. Listen, Robert, we could talk for hours about these things. I think this has been a truly enthralling discussion, and I'm looking forward to having more of them. But this is, in truth, probably one of the most important discussions in terms of its timing and in terms of the relevance to what people may choose to do and what governments and public health officials may need to do in order for us to have a truly safe and a good practice going forward for health. So I want to thank you again, Robert, and I really appreciate the time. I look forward to speaking to you again, and I hope everyone watching has enjoyed this as much as I have. Thank you very much, Robert.
Bye-bye, and thank you for the opportunity and for your interest. And I look forward to learning from your research also, okay? Bye-bye.